Next, we have up Alina Lelic from Beckman Coulter, speaking on streamlined clinical trials, applying rigor and reproducibility in flow cytometry to facilitate therapeutic developments.
Thank you, Christina. All right, thanks everybody for attending. So the goal of my presentation today is to really give you some information and hopefully practical guidelines on how you can apply rigor to your own flow cytometric analysis to facilitate therapeutic developments, or in essence, your own scientific endeavors. So just a little confidentiality, but ultimately, how do we translate science to medicine? And a lot of us forget this aspect because we're locked in the labs and we're doing all of our really fun research. So we forget to see the big picture. But the time that we live in today is a perfect example. If we catch up to coronavirus and we just simply look at this advertisement or this report out of current top 60 treatments in development for specifically coronavirus. And if we go to clinicaltrials.gov, what we'll note is that there are currently 997 clinical trials that are involving COVID-19 specifically, that look at either therapeutic developments, of which there's 695 of them, or just how the immune system responds to the virus itself. So ultimately, when we look at the translation of science to medicine, it really goes down to the clinical trials and the evaluation of different therapies so that we can progress them. And if we just look at flow cytometry in clinical trials, what we'll know is that this was just a simple research that was done with respect to the study title. We see that there's 629. So utilizing flow cytometry as a method to analyze the clinical developments or the actual therapies in clinical trials is an important aspect and is utilized heavily. So how is flow cytometry specifically used in biomedical research? Well, flow cytometry basically is an analysis of single cells, and what it does, it characterizes and evaluates any type of a modulating therapeutic intervention. So whatever this may be, whether it's a vaccine, but it does so on the immune system collectively in terms of the phenotype, function, looking at the health and viability of the cell type, as well as activation state and differentiation, which are all collectively important aspects of collectively analyzing the immune response. And what we really want to do is, in terms of longitudinal assays, in terms of the administration of different therapies, such as vaccines or drugs or small and large molecules, we want to look at the comprehensive analysis of the immune system and assess specifically how these modulating drugs are affecting the immune system. Are they doing so in a beneficial manner so that we can utilize them to progress and develop vaccines out of them, or do they not have any effect overall? So ultimately, the question is, how do we ensure that this quasi-quantitative and a qualitative assay, such as flow cytometry, is reproducible? This is where scientific rigor comes into play. And before we go on, we're going to define what exactly scientific rigor is. So scientific rigor is the strict application of the scientific method, specifically pertaining to our experimental design, method, analysis, reporting, as well as interpretation. So overall, and it has to be done so in an unbiased and well-controlled method. And the reason why we want this to be unbiased and well-controlled is so that we can reproducibly generate data for translational purposes. The translational purposes being the development of immune modulating therapies that can in turn treat patient diseases. So we discussed the fact that there is rigor, and we understand that rigor exists and it's a scientific method. But why are we discussing it? And could it be that there's actually a reproducibility crisis that actually lacks the specific methodology? And in turn, that is the actual truth. If we look at the preclinical data that was generated, this is data that was from 2015 and actually spans to date, we've seen that there's 50% of preclinical data that is actually irreproducible. And that is actually an alarming number because of the fact that if you look at the components of reproducibility, all of these aspects are controlled by the experimenter themselves. and they are reagents and materials to the most extreme effect, study design, data analysis and reporting, and actually protocols. So collectively, we can see that various aspects of irreproducibility can be controlled, but why is it that we are still having this alarming number of 50%? So the actual issues of irreproducibility has been addressed in a workshop that has occurred for the last 5 years. And specifically, this workshop looks at recent issues in bioanalysis. And so this is where the global leading biopharma, biotech companies, as well as CROs, and actually regulatory agencies as well, they get together and they meet for a span of 7 days and they look at all the issues in bioanalysis. Last year, 2019, they came up with guidelines for specifically flow cytometry bioanalysis. And within these guidelines, they specified some of really useful methodologies that can be used. So the first one was utilization of ASR reagents. Now, ASR reagents are analyte-specific reagents, and what this means is that they've been assessed for their specificity, and they're used in diagnostics. So they're GMP manufactured, and they come with specific qualifications and QC specifications through the manufacturer. So what this does for the experimenter themselves, it alleviates them from having to do their own validation in the lab. So it saves time and money. The next aspect is determining concentration, specificity, as well as all of these aspects that are associated with reagents. And in terms of flow cytometry antibodies, this is clonality, fluorescent to protein ratio, and immunogen information. Again, if we look at GMP manufactured materials, vendors will provide this information to you. If it isn't, it is your own responsibility to do this in-house and make sure that you publish this data and that it's recorded, and if you are doing multi-site trials, that it's basically consistent. The next aspect that we all know that it's very critical is titration of antibodies, and we'll go into a little more detail later. Now, FMOs for gating, it's a very important aspect. Now, not necessarily if you're looking at discerning specific populations that are easily separated from each other, such as different lineage markers, but if you are looking at module antigens, it's important that you have your FMO so that you can discern a positive from a negative. If you are changing lots or changing reagents, ensure that partial validation is performed. And then when reading out the MFI in terms of the activation markers, such as your CD69, ensure that you are normalizing with MESF beads. So those are just a few specific guidelines that we've gotten from the workshop. But ultimately today, I'm going to give you a really good overview of how you can implement streamlined workflows into your flow cytometry. And really this is gonna depend on 3 aspects, which is sample preparation, where we're gonna discuss limiting reagent variability, pipetting, minimizing pipetting, and automating the sample preparation. The second aspect is going to come from the data acquisition itself, where we're going to discuss instrumentation, calibration of instrumentation, and standardization. And last but not least, is going to pertain to data analysis. So if you look here collectively, this really relates to that chart where you've seen that 50% of reproducibility. So we're going to hone in on how we can help you reduce that number. So with respect to sample preparation and reagents specifically, we've discussed the ASR specifications. And this just goes on to say that if you have the capacity to obtain unique identifiers and QC of the reagents from the vendor, do so, because it's going to ease your life. So ASR specifications, certificate of analysis, GMP, ISO specifications are all going to be documentation that the vendor provides to you, which basically gives you the QC of the sample and eliminates you from having to do so on your own. Please do minimize reagent lot changes. Now, you may not be able to do so through the entire duration of your study if it's 5 years or longer, but you can actually decrease the number of lot changes by talking to the vendor and saying, please lock this slot for me. Assessing reagent stability, unless that's provided by the vendor. Now, a lot of reagents do not have an expiry date on them, so it's going to be on you to validate the stability of that reagent and how long you can utilize it. In terms of, again, GMP manufactured reagents, those will come pre-validated for you. And then last and most important is incorporation of the experimental controls within your actual daily workflows. So what exactly are these experimental controls? They're internally qualified samples that actually contain your antigens of interest. So let's say we are looking at a simple analysis such as TBNK, and you're looking at your CD3, CD8. So this internal sample is going to be a leukapheresis pack internally that you've frozen down, and you've run 20 times and specified the positive ranges as well as the variability within it. Some manufacturer standards are available, such as Cytotrol and Immunotrol, and they basically take away the necessity for you to validate the reagent and specify the ranges. And they look like this. So in cases of utilizing the Cytotrol, so what you're going to do is run these assays and run these cells every single time you run your experiment. And you're going to ensure that your cocktailing, antibody cocktailing that you prepared that day, is effectively accurate, such that you're not missing an antibody. And it's going to also ensure that your pipetting errors are not present, such that you've added too little of an antibody and now the CD3 proportion has decreased due to the pipetting error. And it's also going to allow you to see if your instrument stability is actually, in essence, good, and that your instrumentation hasn't incurred some sort of instability. But ultimately, what this is going, collectively, the internal experimental control is going to validate the fact that your experiment is good, and that the biological changes that you're actually seeing from sample to sample are real and not a byproduct of any errors. So we talked about the reagent variability in terms of the fact of cocktailing. So when you cocktail your antibodies, you can definitely have issues such as adding the wrong antibody, but not just that, but adding too little of an antibody, or sometimes just overall forgetting it. But before we actually get to the cocktailing, we're just going to briefly touch upon the panel design. Panel design is a tedious process, especially when we increase the number of conjugated antibodies in our panel. What this means is that we're going to have to select our antibody clones, select the dyes, and verify the panel performance. And every time we go through these iterations and select our FMOs to ensure that the panel is indeed giving us biologically relevant data that mimics what we see in single color stains, we're going to have to validate the panel. Once the panel has been validated, we're going to optimize the preparation of the protocols and then transfer the assay into the lab. So the entire protocol here takes a lot of time, and ultimately the entire process itself can lead to variability and error, specifically when it comes to antibody titration, cocktailing, and antibody validation themselves. All of these three aspects themselves can actually contribute a lot to variability, but ultimately they lead to loss of money and time, which is not something that we want to necessarily focus on, especially if we're looking at developing therapies. We want to make sure that we're not delaying due to the methodology. So, is there an alternative method to the liquid cocktail antibody workflows? And it just so happens that the various vendors right now contain these dry and optimized ready-to-use cocktails. These dry and optimized cocktails are pre-designed with specific antibodies of your choice that you pre-selected, or the ones that are currently existing off the shelf. And all they require you to do is to basically order them in, validate the panel performance and test the lot itself, and then transfer the assay into the lab. So collectively, these panels eliminate the titration, the panel design, and the actual antibody validation, because all these aspects have already been done for you. So this is how we increase the efficiency, and this is how we implement the fact that we develop these therapies much quicker. So this is just a Beckman Coulter immune cell phenotype and characterization panel. It's called the Duraclone IM antibody. And these specific 8 panels were designed with experts in the field that came to Beckman Coulter and said, we are doing a global study in transplantation, and we need a panel that's going to be stable, room temperature stable, that we can basically ship to all of the different multi-sites and ensure that we have robustness and we have reproducibility. So they came together and they designed these beautiful 8 panels that comprehensively analyze the immune system, whether it's dendritic cells, whether it's just a basic phenotype you can see here by the markers. And if you look at the panels here, this is just an example of the 3. So specifically when looking at the Treg tube, it characterizes the Tregs based on your 25, FOXP3, as well as various 39 and Helios. What's important to note is that this panel can be utilized as a base with additional drop-ins. So let's say that I wanted to, in addition to just assessing my T cells based on my Tregs based on FOXP3 and Helios, I wanted to actually look at the memory phenotype of my cells. I can additionally add CD127 and CCR7 and look at the actual proportion of memory cells. Likewise, in terms of the granulocyte tubes, this is your base panel that you can, in addition to having the base panel and depending on the configuration of your flow cytometer, you can add additional antibodies as drop-ins. So to additionally analyze cells of interest. In addition to the phenotyping panels, there are indeed functional characterization panels, and I'm just showing 4 here. These are the T activation panels, T helper cells, monocytes, and basophil. And this is just a simple representation of the data that you obtain. And last but not least, we do have the rare event antibody detection, which is heavily utilized in clinical trials that are looking at CLL, multiple myeloma, or ALL trials to ascertain how many of the rare events are present and the prognosis of different patients on therapies. So collectively, these panels have been assessed for stability. And with respect to comparison to the liquid antibody cocktail, you could see in terms of the overlay of the data here, there's no differences between the liquid or the pre-formulated. The brightness is preserved across all dyes, and there's no changes in compensation. Why is this important? Well, it's important in the sense that you can see that the stabilization of the actual cocktail did not contribute to changing of the actual brightness or any stability of the dye itself. So even tandems in this case are stabilized. If you look at temperature sensitivity, you can see that even in extreme temperatures, these dyes have been preserved. And basically the entire panel is room temperature stable, so it eliminates the necessity for refrigeration. So you do have options to utilize these dry panels to ensure rigorous methodologies when it comes to your reagents and to ensure the best reproducibility when you are assessing clinical trials and patient responses to specific therapies. Now, how else can we actually reduce experimental variability? One of the aspects is to utilize automated workflows. So what I'm showing you here is just simply a automated workflow that is utilizing a automation handler along with the flow cytometer that is doing the sample preparation, antibody cocktailing, as well as staining, and then integrating this with flow cytometry. It's delivering the prepped samples that have been washed within a centrifuge and delivers them to flow cytometer and analyzes the data. This data can then be, this analysis can then be integrated with specific utilization of API to basically allow you to push the data out to a cloud for the additional analysis. Now, what's important in terms of the actual workflows is that the automated workflows do reduce experimental variability. And what I'm showing you here is a graph and the analysis of the T cell phenotype in terms of the intra-assay reproducibility with respect to the mean phenotype when we just looked at the T cells and specific populations of the T cells, and then the percent CVs. So what you see here is consistently less than 20% variability among the 96 replicates, and even in the cases where the percentages of the events were less than 2%, where you would expect the variability to increase significantly, which in turn is the case. So what you're consistently seeing here is very good reproducibility. Now that we've acquired the data and we trust that the data has been accurately acquired and we know that our instrumentation has been optimized, we're going to go on to data acquisition. So when we look at the data acquisition itself, it is important that all flow cytometers, it's important to understand that all flow cytometers are inherently different, whether that relates to optics, fluidics, detectors, and electronics. And every instrument has to be evaluated. And so in such, whether you do it through the vendor qualification, IQOQ, you still have to ensure that you understand the quantum efficiency of the detectors, the electronic and the optical background, as well as the spillover spreading matrix, so that you can understand the sensitivity of your instrument and its best utility. In addition to that, if you are going to be submitting the data to FDA, make sure that you do have 21 CFR Part 11 compliance tools that ensure that the electronic records and signatures are considered trustworthy and reliable. So this is just going to expedite the process of the submission. Now, with respect to optimizing your instrument settings, this is important for every single flow cytometer. So this is where we're going to evaluate our flow cytometer and understand where the signal resolution is best. And we're going to ensure that optimization of instrument setting is going to provide us best signal resolution. Why this is the case? The reason for it is so that we consistently acquire good data. And what I mean by that is so that we consistently know that the changes in our sample acquisition is not due to this instrumentation, but actually due to the biological differences. The next aspect of instrument qualification is standardization. Whenever you are doing a longitudinal assay or if you're doing clinical trials, this is a very critical component of ensuring that biological differences are real. So what we're going to do is we're going to utilize an MFI and we're going to ensure that the MFI is utilized to set the target settings on a daily basis to ensure that we are performing to ensure that we are utilizing the same settings throughout the duration of the study. Now, once you've optimized and standardized your settings, you're going to go on to verify the experimental settings as well as the compensation matrix. And what I'm going to show you here is just a simple data analysis that was done on the Cytoflex that contains a live dead cell, CD3, as well as CD4 and CD8. So just a simple subset analysis. And if you look at the proportion of CD3 positive cells, you're looking at 99% positives, which goes on to show the CD4 and the CD8. So now, let's say you were a little bit alarmed by this number because you know these are your PBMCs. So the total percentage of CD3 shouldn't be 99. So you go on to verify your compensation matrix utilizing your single color controls and ensuring that everything is accurately done. And you adjust your compensation matrix to, in the end, obtain a CD3 positive population, which is indeed 79 positive, which goes to show that the compensation matrix that you first originally identified was actually overcompensated, and the population of CD3 was reported erroneously. So if you do utilize unoptimized settings and if you don't fix the compensation, you will, you might actually report out erroneous data, which could be detrimental. So once we have analyzed the instrumentation and we've acquired, optimized, and standardized settings and we've validated everything, the next aspect is the data analysis. And so data analysis can be very challenging. And I'm just showing you a couple of biplots here, but the more high dimensional our data is, the more challenging it is to analyze it. And these challenges simply go from the fact that there's too many plots to analyze. You have variability in the manual gating in terms of how I do my analysis with respect to how you do it, or which population of interest I go to gate first. And then ultimately user bias, you know, what I consider a positive, where I consider the gate to be set appropriately, you might not necessarily think that's the appropriate setup. And ultimately, this leads to irreproducibility. And you saw on that irreproducibility components that data analysis does contribute a lot to the actual inability for us to reproduce the data. So what if we could take all of these biplots in our high dimensional data and reduce it, but still contain the complexity of the data, but eliminate the bias? And what if this reduction looked like this beautiful plot, beautiful Visney plot, that still, as I said, contained the complexity and contained the ability for you to see specifically where each of the antigens of interest were labeled and their expression levels. So what you're looking at here is a Visney map that's reduced all of that complexity of the data, separated your events based on like similarities into these islands that then distribute into CD8 positive T cells and probably monocytes or whatever else, that you can technically utilize the z-axis over here to look at the expression. In addition to that, you can do further analysis and reduction of the complexity of the data by looking at clustering, and then specifically looking at how the different clusters differentiate based on the expression of different antigens. So collectively, you can actually utilize these unsupervised and supervised machine learning algorithms, such as the ones I identified here, Visney, FlowSum, Citrus, and Spade, to either reduce dimensionality first, cluster and organize your data, or look at the expression levels within those actual specific clusters. And what that's going to allow you to do is reduce the complexity of your data, ultimately, eliminate the bias of manual gating, because you're allowing a computer algorithm to go through different iteration of data analysis and do so without the bias of you setting the gates. And ultimately, what we want to do here is objectively discover new populations that are going to give us potential biomarkers that are associated with different therapy evaluations.
And just real quick, I want to let you know you have about 4 minutes left.
Thank you. That's perfect. Okay, so ultimately we want to increase the reproducibility of the data, and the significance of rigor in our clinical research is through the different sample preparation, acquisition, and analysis, is ultimately to unbiasedly and well-controlled evaluate the immune therapies so that we can actually develop immune modulating therapies to treat patients. So rigor is important because it allows reproducibility and efficient ways to do so. And so that is all. Thank you very much. And I'll take any questions that you may have.
Wonderful. Thank you so much for that wonderful presentation. So we have a couple of questions. One is from Jay Wen, who wanted to know if you have any comments on determining the in-use shelf life or stability, after opening, I'm assuming for the Duraclone reagents.
Okay, so the Duraclone reagents themselves actually come with a pre-validated shelf life stability, and it's currently at 18 months. So as we progress with the evaluation of the shelf life, this keeps on increasing. But as of now, we have an actual documentation that you can ask for, and we can give it to you.
Perfect, thank you. And then I actually have a personal question about compensation and modifying compensation and its impacts on rigor and reproducibility. So when we're taking a look at any of our data, I know personally we don't advise manual adjustments to compensation. So I'm wondering how you suggest checking that compensation, and how do you validate that that's reproducible if you make any adjustments there?
Okay, so ultimately the compensation adjustment, so prior to you doing any compensation adjustment, the most critical aspect I would say that's going to ensure that you have good compensation is optimize settings. So when you optimize your settings, you're going to basically allow, once you've done the analysis, you can run your single column controls and see how the actual data spills with different, and open up all the channels. So once you do the compensation calculation with your optimized settings, what you'll do after that is utilize your single color controls, and when the optimized settings have been generated, when the optimized settings have been generated, you will notice that there's going to be minor shifts in the compensation that you actually have to adjust. Now, when I say minor shifts, what I mean by that is just like those slight movements of the populations. And how you can ensure that what you're doing is reproducible is basically applying that same compensation matrix and applying it to the rest of your, so taking, I don't know if this is done, how you can do this on different instrumentations, but I know specifically within the Beckman Coulter instruments, you can save that compensation matrix and apply it to subsequent analysis. So basically, anytime you make any modification, it has to be done. You have to optimize it in the beginning and then consequently do all of the analysis on the same matrix.
Great. And would there be any way, because I know, you know, as a follow-up to that, as we only have about 30 seconds left, you know, I think a big part is controls, right? And the appropriate controls to have the correct compensation.
Okay.
So, you know, it would be just an important thing to note there as well for reproducibility. And that was all the questions we have. So that was a really wonderful talk, and thank you so much. Perfectly on time. So thank you very much for this.
Thank you, guys.